See, in the field of population genetics, the name of G.H. Hardy and W. Weinberg is taken with great respect. The reason is that these two persons independently gave a very important principle regarding the fate of gene and genotype in a sexually reproducing population. Hardy was a British mathematician and Weinberg was a German physician. These two persons independently explained that the frequency of gene and genotype will remain constant generation after generation in a large randomly mating population, provided if this population does not experience mutation, selection, random genetic drift, and migration. If these conditions are met, then the gene and genotype frequency will not be undergoing any change in ensuing generation or incoming generation, or that will remain constant generation after generation. Before I explain this principle in much detail, let us have some idea about the population, because this particular principle is based on, or that is mainly focused on, the population, particularly the sexually reproducing population. A population is a group of individuals that includes all the members of a species living in a specific area during a specific period of time. So, group of individuals which are interbreeding one and which belong to a specific species, they form a population. In fact, the population is a unit of evolution. So the smallest unit of evolution is a population. This is another important thing one should know. That is, evolution that occurs means at micro level, the evolutionary events occur in the population. So the change of an allele frequency over time within a population is known as microevolution. We also talk about Mendelian population. A Mendelian population is an interbreeding group of organisms that share a common gene pool. So the individuals which form a population, they are capable to interbreed among themselves, and they have a specific gene pool, so that forms a Mendelian population. This applies in case of plants, animals, fungi, so all the diploid forms of organisms which can reproduce sexually, they form a Mendelian population because the Mendelian rules of inheritance are followed in them. Let us see what is a gene pool. Actually, the sum total of all alleles that forms the gene pool. So the gene pool is the total number of alleles present in a population. And the concept of a gene pool usually refers to the sum of all the alleles at all the loci within the genes of a population of a particular species. Then let us come to the equilibrium. What is Hardy-Weinberg equilibrium? The Hardy-Weinberg equilibrium helps us to know if a population is evolving or not, because Hardy and Weinberg talked about an ideal population. That is, an ideal population will not be undergoing any kind of change, because that population, if it is quite large one and if it is randomly mating one, and if mutation doesn't occur in it, selection does not operate in it, gene flow is totally inhibited, that is, individuals are not moving from one place to the other, or there is no exchange of gametes between the adjacent populations. In such case, and the population is not getting reduced to a very small size because of any reason, so in that case, that population will not be experiencing any genetic change and it will remain constant. But in reality, this does not happen. So if a population is in Hardy-Weinberg equilibrium, then the allele frequencies will remain constant over time. Now suppose that we can actually understand this aspect in a more better way by considering a single gene locus. Suppose there is a specific gene locus which is being represented by two alleles, that is capital A and small a. Because of the presence of these two alleles, three different genotypes will be possible, and those three genotypes will be capital AA, capital A small a, and small a small a. Now if we consider that the frequency of this capital A is P and the frequency of small a is Q, then this P plus Q will be equal to 1. And if we consider the genotype, that is capital AA, this will be represented by its frequency, that is P square. Capital A small a, that is heterozygotes, will be represented by 2pq, and small aa, that is the third genotype, the third type of individuals, will be represented as q square. And the total of these three genotypic frequency, that is p square, 2pq and q square, will be equal to 1. Now we can have better idea about this mathematical derivation. See, once again, the same thing I would like to repeat, that a Hardy-Weinberg equilibrium could be seen in a population if that population does not experience mutation at all. A random mating is occurring in that population if it is quite large in size, and the laws of probability function in that specific population. There is no exchange of genes, that is migration is not there at all, and selection does not operate, then that population will be expected to remain in equilibrium. Now suppose we are considering a single gene locus, and as I just told, that frequency of capital A is represented by P, Q is the frequency of small a, then P plus Q will be equal to 1. Now suppose two types of individuals, that is the male and female are there, gamete formation will be occurring in them. Male will form two different kinds of gametes, that is capital A and small a. In case of female also, two different kinds of eggs will be formed, that is eggs containing capital A allele and the other one is small a allele. And there will be random fertilization of these gametes, that is eggs of these two types will be randomly fertilized by sperm of either of the types. So if the egg containing capital A allele gets fertilized with a sperm with capital A, then you will have individuals with genotype capital A, and their frequency will be P into P, that is P square. In other case, if the egg with capital A allele gets fertilized with a sperm having a small a, then the individual form will be heterozygous, and here P and Q, that will be the frequency of this. Now since two such combinations will be formed, that is capital A small a, if a small a containing gamete gets fertilized by a sperm having capital A, in such case also a heterozygous individual will be formed, and here again PQ will be its frequency. So all heterozygotes out of this particular crosses will be 2PQ actually, and then the individuals with small a will be formed when an egg containing a small allele gets fertilized with a small a sperm, then in such case the frequency will be Q square. So here the frequency of genotype will be P square, 2PQ and Q square, and their total will be equal to 1. Now this is an excellent diagram which actually helps to understand so many things. As I told in the beginning that P plus Q will have to be equal to 1. So if we see in the lower side on this x-axis, the frequency of P is shown starting from 0 to 1. And just on the upper side you can see the the figures are 0 to 1 but in just uh, opposite direction. So what we can see here on the upper side, the frequency of Q is denoted. Now if in a population the frequency of uh, capital A allele, that is P, is zero point 0.5, then the frequency of small a allele, that is Q, will also be zero point 0.5 because total will have to be 1. If you consider the frequency of capital A zero point 0.1, in such case the frequency of small a, that is Q, will be zero point 0.9 because total will have to be 1. So if you take the frequency of small a on the upper side, that is zero point 0.7, then the frequency of this Q will be zero point 0.3, that is the total becomes 1. So this way you can have the idea about the frequency of capital A and small a are P and Q. Now these three lines, they represent about the genotype. Like Q square, this represents about small a types. 2pq, that indicates about the heterozygotes. Those individuals whose frequency is denoted by p square, they are in fact capital A, capital A. So what I wish to tell you, that if the frequency, if you see here, if the frequency of, suppose uh, this Q is zero point 0.8, I am starting from this 0.8. If the frequency of Q is zero point 0.8, then Q square will be 0.64, means you just start coming down, make a line, because if the frequency of Q is zero point 0.8, the frequency of P will be zero point 0.2, so you make a straight line connecting this zero point 0.8 with zero point 0.2, and you will come here because this line will be joining with this place. So it will be exactly 0.64. So the frequency of Q square, that is a small a, will be 0.64, and that could be seen on the left side of this line. On the left side of this line, you see these are the digits like 0 0.2, 0 0.4, 0 0.6, 0 0.8 and then 1. And on this right side, just opposite situation will be there. But I am considering here, if the frequency of Q is zero point 0.8, then Q square will be 0.64, and that could be seen on the left side of this line. Then you come downward, come to this place, then 2pq means heterozygotes are being represented. So this frequency will become 2 into zero point 0.8 into zero point 0.2. So that will be 0.32. So you will find figure 0.32 here. And if you come downward, then you will see that the line will be touching here, and since the frequency of this uh, P is zero point 0.2, so it will be 0.04. So exactly 0.04 will be the frequency of P square. So this is one excellent line diagram through which the expected values of three different genotypes could be known. Because if you come in the center of it, if the frequency of P is zero point 0.5, then P square and Q square, both values will be exactly 0.25. And then the heterozygotes will be 0.5. Okay, because 2 into 0.5 into 0.5, this value will exactly be 25, 0.5. So you can say that the maximum expected frequency of heterozygotes will be 0.5, maximum frequency of heterozygotes. So this line diagram helps us to understand the expected values of these three different genotypes. Now I am going to take up an example to see that how we shall test that a specific locus of a population is in Hardy-Weinberg equilibrium. This example will be of much significance because it will give holistic idea to analyze that whether a specific locus is in equilibrium or not. And for this purpose, I am going to take up the example of Yemen blood group. Although this Yemen blood group is not of much pathological significance, but we can actually test the human population whether a person is of M blood group or N blood group or is heterozygous, because by using two uh, antisera, that is antisera or antibodies M and N, one can analyze human population, and this Yemen blood group system could be analyzed, could be tested. So it's quite different from ABO blood group system, and it does not cause any immunological reaction, so it is not of much significance. But in the field of genetics, this example is quite often taken because the entire human population could be divided into three different blood group types, that is M, MN and N. So, but here the very important and important thing is that those individuals whose blood group is M, they are actually genotypically MM types. It means their mother and father both actually contributed M allele to this individual. And those whose blood group will be MN, they have received one allele in the form of M from one of the parents and the other N allele from the other parents. So they are actually heterozygous. It is one of the best examples of codominance because both alleles show their expression. And individuals with the N blood group type, they are genotypically NN. Now suppose you have randomly selected 100 individuals from a specific area, from a specific place, and you tested this Yemen blood group in them and found that 40 individuals out of 100 are of blood group M. Then heterozygotes, they are also 40 in number, and you got that 20 individuals are of blood group N. So we want to know that whether this distribution of blood group types is in equilibrium or not. So you got this number. You actually observed this number by using antibodies. You tested 100 individuals randomly from an area and you got this number. So next step will be to find out the frequency of M and N in the population. Only then you can calculate the expected number of individuals, because this number you have observed, so you have to find out the expected number, and for that you will be calculating the frequency of M and N alleles. So the frequency of M allele, which can be represented by P, will be equal to 40 plus 40 plus 40. Why I am taking 43 times? Because 40 individuals are of blood group M, and each individual will carry two chromosomes. So both chromosomes contain actually M allele. So 40 will be added twice. So 40 plus 40. And then heterozygotes, only 40 chromosomes will be containing M. Remaining 40 will have the N types. So that is why I am considering 40 here. And since 100 individuals have been analyzed, so 200 alleles or 200 chromosomes have been actually observed. So this figure will be coming zero point 0.6. So 0.6 will be the frequency of P, that is M allele. Now the total will have to be 1. So the other one, that is N allele, its frequency will be 0.4. Or you can consider 20 plus 40 divided by 200. So this will come 0.40. So once you have calculated the frequency of M and N in the population, you can find out the expected values or expected number. That expected number will be obtained by doing this, that is P square into number of individuals analyzed, that is capital N. So this zero point 0.6 is the frequency of P, that is zero point 0.6 square into 100, because 100 individuals have been observed, so this comes 36. 2pq into n, so when you will multiply 2 into 0 0.6 into 0 0.4, you will find, and 100, then you will get 48. So 48 will be the expected number of heterozygotes. Q square into n, that will give you the expected number of n types, and by using this formula you will be getting this number 60. So you actually expect 36 as M types, 48 heterozygotes, and 16 that is N types. So we have to know whether the observed number and expected number, they show very close, closeness in them, or there exists significant difference between the two. So when you have to analyze the difference between observation and expectation, in such case the statistical test which is applied is chi-square test. So we, in the next step, will be going for chi-square test. Actually, this chi-square test is always performed to see the difference between observation and expectation. It is a kind of non-parametric test, and this is computed by using a formula, that is summation observed minus expected its square divided by expected. So using this formula, you can find out the chi-square values. Now the chi-square value of the first case means observed value is 40, expected value is 36. So 40 minus 36, its square will be taken. So that will be 4 square and 16. This 16 will be divided by 36. You will get the value 0.44. In the next case, 40 is the observed number and expected number is 48. So 40 minus 48, it will be minus 8. Its square will be 64 divided by 48, and this value will be coming 1.33. Likewise, here in this case, 20 minus 16 will be 4. Its square will be 16, and divided by 16, this value will be coming 1. So when you will add up these values, final chi-square value will be 2.77. Now, once you have got this value, chi-square value, this is actually calculated value. Next step will be to find out degree of freedom. And in this case, degree of freedom will be calculated by using this formula, number of phenotypes minus number of alleles. Since there are 3 phenotypes and 2 alleles are there, M and N, so this degree of freedom comes 1. And at that degree of freedom, at 1 degree of freedom, at 5% level, the table value is 3.84. So this is actually critical value. Critical value represents the table value, chi-square table value, and at 1 degree of freedom, this value is 3.84 at 5% level. So what we have to see whether our calculated value is more than this or not. Our calculated value is less than this 3.84. It means we accept the hypothesis. We accept the hypothesis means we accept Hardy-Weinberg equilibrium hypothesis, that is the present population is in equilibrium, Hardy-Weinberg equilibrium. So here we actually probability is less than, it is probability is more than 0.05. It means occurrence of this event is more than 5%, and accept the null hypothesis, that is the genotypic frequencies on this locus in this population exist in Hardy-Weinberg equilibrium. So we can say that this locus, Mn blood group locus of this specific population is in equilibrium. So in fact, it is because there is insignificant difference between observation and expectation. And when we find that the calculated value is more than the table value or critical value, then we reject the hypothesis. We can say that significant difference exists between observation and expectation. So by using these steps, one can be sure whether a population is in equilibrium or not. Okay.